Okay, in this session we're going to cover some genetic concepts that are central to the understanding of cancer, and this is looking at multi-hit models, looking at clonal evolution, and looking to some extent at the order of events that result in very heterogeneous appearing cancers. Okay, so initially we're going to cover the monoclonal nature of cancers, and the monoclonal nature of cancers it basically suggests that cancers come from a single mutated cell, irrespective of how they look. And there's data that appears to be the opposite of this, so prostate and colon cancer sometimes develop in a polyclonal manner, which is multiple independent cancers, and some tumours develop with a mixed phenotype, so adenocarcinoma with squamous cell carcinoma mixed together. So these can appear to be multiple different tumours in the same place. The vast majority of cases, however, a single mutant cell leads to a tumour, and that tumour can appear to be very heterogeneous or can appear as multiple independent cancers. So in this session we're going to look at this monoclonal nature of tumours and look at the sequence of events that leads to cancer and look at examples where this might not be true, but in 99% of tumour types this is true. So if we look at what is currently accepted as the standard way that tumours form, here is a whole series of cells and one of them develops a mutation. That cell develops multiple subsequent mutations and that will give rise to multiple subpopulations within that tumour that can appear to be highly heterogeneous. This could be a bit of squamous and a bit of adenocarcinoma and all different phenotypes in between. Whereas the alternate hypothesis, which has been disproven in most cases, initially it was thought that maybe multiple cells within a tissue each individually become mutated and they form to produce this mass of cells with very varying phenotypes, giving the heterogeneity. Now what we now know is that this heterogeneity comes from different populations of cells. You initially form a tumour and then maybe this cell here has different mutations to give it a different morphology and you end up with a heterogeneous tumour. Now there's only a few examples that you should be aware of which are genuinely polyclonal, and that would be where multiple independent tumours form completely independently of each other. So for example in hereditary breast cancer you can have multiple independent tumours in one breast. Similarly with prostate cancer, you can have multiple independent tumours within a prostate which are genetically unrelated to each other. But these are, and also in colon, particularly if you've got a genetic predisposition to colon cancer, you can have multiple independent colon polyps which can form multiple independent tumours. But these are very much the exception, and in most cases cancer develops as a single tumour at a single site originating from a single cell. Okay, so there's some very good evidence to prove beyond doubt that tumours are monoclonal in origin, and this evidence comes from the genetic phenomenon of X inactivation. So if you were to hypothesize that cancers were polyclonal in origin, you can use cancers in females to test this hypothesis. In females, you should all know that one X chromosome is randomly inactivated during embryogenesis so that in adult tissues each individual region of tissue has either the maternal or the paternal X chromosome inactivated. So this is random X inactivation. So large M is a maternal X chromosome that's active, large P is paternal X chromosome which is active. What we find is in females when tumours develop, we look at if you look at the adult tissue, you've got a patchwork of maternal cells expressing that particular X chromosome. But when we look at individual tumours that form, they almost always have all the same X chromosome expressed. So in this case, a tumour might have arisen from this individual cell here, so therefore all the subsequent cells have got the paternal X chromosome expressed, never the maternal. So when we look at an individual tumour from a female, we see the same pattern of X inactivation in all cells within that tumour. Therefore, we can be absolutely sure that those tumours are monoclonal in origin, even if they appear to be polyclonal. Okay, so once we've accepted that tumours are monoclonal in origin and the heterogeneity that is observed is due to random mutations giving different subpopulations different behaviours, we need to look at how that can develop. And this Heterogeneity is developed via the multi-step nature of cancer formation, whereby a single cell develops a mutation, and we're going to look at this model in detail, Vogelstein's model of colon cancer formation. So a single normal epithelial cell acquires a mutation. In this case, it is the APC gene, Adenomatous Polyposis Coli. So that's the first hit. Then you've got a second hit, and then subsequent mutations occurring. Now, when these subsequent mutations occur, they only occur in a minority subpopulation. So this KRAS mutation might have only occurred in 1 out of 1 million cells that are in this early adenoma. And then this SMAD4 mutation might have occurred in only 1 of the 1 million cells that have got a KRAS mutation. So you end up with huge amounts of heterogeneity across a tumour, but the tumour itself appears very, very heterogeneous as dominant clones outgrow, and then we end up at the end of this process with a fully fledged cancer which has got all of these mutations, but there might be lots of other pre-malignant cells which have 1 or 2 or 3 of these mutations but are not yet fully fledged cancer, and that's why we end up with a highly heterogeneous cancer. And other tumours achieve this, but using different genes in a different order of events. So this can be summarised here. So this is showing a typical multi-step tumourigenesis of, in this case, colon cancer. And we can start off with a bunch of cells which are metaplastic. They're not yet malignant, they're just genetically abnormal. And what we find is if we can take a cross-section across a tumour and genotype individual bits of the tumour, we can find a series of events that results in maybe 5 cancerous clones of cells, all genetically related to each other, all genetically related to, say, this cell here. So all of these 5 or 6 clones have acquired a whole series of genetic mutations, but they all form one individual cancer. And they've done it in different ways. So this one has lost chromosome 17p, which is where the p53 gene lives. So at this point, it's p53 null. And then we've got other mutations that have occurred, and it becomes aneuploid, and that's one clone of cells. We can follow this clone of cells through here. So a different clone of cells might have lost p53 and lost chromosome 9p, so the CDKN2A, tumour suppressor gene, and then subsequently lost other things. 13q is your retinoblastoma gene, and then something else has happened, 18q, probably SMAD4, and we end up with a cancer clone. And you can follow the same sequence of events happening in other cancer clones. So you can end up with a very, very heterogeneous cancer with different regions of this cancer all being genetically related to each other, but they've sort of got there in slightly different ways, but using the same genes but in a different order. And this tends to be what happens. You start off with a large pre-malignant lesion, which is all of this region here, and so all of these might be pre-malignant, and then you get clones of cells which are fully fledged invasive cancer that have got that final mutation that's required to create a fully fledged cancerous clone. So what are these genes that I'm talking about here? You know, p53, CDKN2A, and so on. And these fall into 2 different families, tumour suppressor genes and oncogenes. The tumour suppressor genes normally either suppress growth, so p21 or CDKN2A. They can repair DNA, such as HMLH1, which is a mismatch repair gene, or they can promote apoptosis, such as BAX, which opposes BCL2, part of the apoptotic pathway. So tumours really benefit from inactivating tumour suppressor genes, whereas tumours benefit from activating oncogenes. So these typically promote growth, like KRAS and epidermal growth factor receptor, or they oppose apoptosis, such as BCL2 and AKT. These genes are all typically activated in cancer. And this slide shows where many of these genes are, and I've highlighted in red tumour suppressor genes and in green oncogenes. So here we've got a mitogen that binds a mitogen receptor, so this could be epidermal growth factor binding epidermal growth factor receptor. Downstream of this is RAS. RAS can become activated by mutation. This signal is passed on to the MAP kinase cascade, and each of these can become activated by mutation. So we're looking at RAF, MEK, ERK, those sorts of proteins. This switches on c-MYC, and MYC can become specifically activated by gene amplification or translocation sometimes, so we get excessive amounts of MYC produced. This switches on cyclin D, which is an oncogene, so all of these are oncogenes because they're green. P16 can become inactivated. P16 inactivates P16 inactivates CDK4, so a tumour would really benefit from losing P16. And then downstream of cyclin D, we've got the PRB protein, retinoblastoma. Tumours really benefit from losing that, and the function of PRB is to hold on to S-phase promoting S-phase. E2Fs are crucial for S-phase promotion, so tumours can overexpress E2F transcription factors, and that will drive the cell through to S-phase. So what we find is that tumours will activate usually one of these oncogenes in green, or inactivate one or more of these tumour suppressor genes, but they don't need to do all of them. Once they've done something like loss of PRB, they don't necessarily need overexpression of E2Fs, because those two genes are doing exactly the same sort of function. That, you know, losing PRB is phenotypically the same as gaining E2F. So the tumours will have one or more of these mutations on this pathway, but they don't need all of them. And different tumours will achieve activation of this pathway by one or more oncogene mutations or one or more tumour suppressor gene activations, but not all of them. And different tumours will select a different way of doing that. So just a reminder for you of what retinoblastoma protein does. It's the guardian of the R point. So retinoblastoma regulates signals to allow cells to go from early G1 through to late G1 and into S phase. So hypophosphorylated retinoblastoma sequesters E2F transcription factors. These transcription factors drive S phase. So after the R point, the E2Fs are released because PRB is hyperphosphorylated, and that hyperphosphorylation event is via cyclin D activating cyclin-dependent kinase 4 and cyclin E activating cyclin-dependent kinase 2. They will phosphorylate retinoblastoma, and that will cause E2F release. And we can see this on this pathway here. The key point here is we are getting activation of the S phase, the activation of cyclin E and via its CDK. Now the G1S cyclin here is cyclin E. And this slide, this part here shows what is happening. This is the retinoblastoma protein, this is the E2F transcription factor in early mid G1. We then get phosphorylation events put on there by cyclin D activation of CDK4. We then get cyclin E activation of CDK2. That hyperphosphorylates PRB. We get release of the E2F transcription factors, and those E2F transcription factors in S phase are bound to chromatin, switching on S phase relevant genes. Now we can get loss of the retinoblastoma gene, and that will promote cell cycle progression. We can get that loss by gene deletion or gene mutation, but we can get exactly the same effect by overexpressing E2F transcription factors, or we could get exactly the same effect by being infected with human papillomavirus and its E7 protein, which inactivates the retinoblastoma protein. So you can get multiple different ways to have this event of G1 to S phase transition abnormally in cancers. And basically all of the mutations, all of the oncogenes and tumour suppressor genes that I've mentioned, all will promote this. So everything that I've mentioned on this slide, either loss of PRB, gain of E2Fs, infection with this virus, will all promote G1 to S phase transition in one way or another. We can also get gene mutations in genes that affect cell cycle related to the p53 pathway. So if we look at this pathway, radiation or anything else that causes DNA damage will activate this series of kinases, which will phosphorylate p53, and p53 acts as a transcription factor, switching on the p21 gene. The p21 protein will naturally bind and inactivate CDK, cyclin-dependent kinase, and cyclin complexes. So here is the cyclin-dependent kinase, here's the cyclin that's activating it, and when p21 is bound to it, this is inactive. And I've just moved the screen so you can see the detail down here. So we can see p53 is stabilized by DNA damage. In the stabilized form, p53 induces p21. p21 stops the cell cycle by binding to the G1S and S-cyclin CDK complexes, so stopping the cell cycle at G1S and S phase, that's cyclin E and cyclin A. But p53 also induces other response genes to induce apoptosis at the same time, so induces BAX to overcome BCL2 and induce apoptosis, induces Tumour, which is a BH3 only protein, to oppose BCL2. So what p53 is doing here when it's normally functioning is stopping the cell cycle and inducing apoptosis, and this is why tumours gain a benefit from losing p53 expression. If p53 is non-functional, we don't get cell cycle arrest and we don't get apoptosis. Now, quite often the first event in tumour formation is the loss of a tumour suppressor gene, so it's worth revisiting or visiting for the first time Knudson's two-hit hypothesis of tumour suppressor gene inactivation, because tumour suppressor genes are on there are two copies of almost all tumour suppressor genes, and they are recessive in their activity. That means you've got two independent copies of the gene, and therefore you need, to have no tumour suppressor gene activity, both copies of the gene need to be inactivated either by gene mutation, a point mutation, gene deletion, epigenetic silencing, something like that. So there are two copies of a tumour suppressor gene. So generally, loss of a single copy of a tumour suppressor gene has no effect. Now, there's one exception to that, which is p53, where it does have an effect, and we'll come on to that at some other point in the module. In contrast, oncogenes are dominantly, the mutations are dominant in activity, and therefore only a single activating mutation is required. So we've already seen this Vogelstein model. The first hit in colon cancer tends to be the APC gene, and you get the first hit either if you have got a hereditary colon cancer, you could have the first hit being a hereditary mutation in the APC gene, and that causes familial Adenomatous Polyposis Coli, FAP. That's a hereditary colon cancer syndrome. In most cases, people who are not genetically predisposed, the first hit is a somatic mutation of the APC gene. Okay, so once a patient has either inherited this mutation or a somatic cell has acquired this mutation, that's your first hit. The second hit is another mutation on the other copy of the APC gene, so that at this point, both this patient and this patient are in the same situation, the APC gene is inactivated, and that results in the formation of an aberrant crypt, and that is the early stages of tumour formation. We then get genomic instability, which we can talk about at some other time. Then we get activation of KRAS, which is an oncogene. We then get SMAD4 mutations, loss of p53 tumour suppressor gene, and that leads to cancer formation. So it's this early event that's really worth looking at about how tumour suppressor genes can become inactivated, and this two-hit model. Now, many of the major tumour suppressor genes that we know of were known about because they cause hereditary cancer syndromes. So, for example, the BRCA1 and BRCA2 genes cause hereditary breast cancer, and patients develop either unilateral or bilateral breast cancer, whereas sporadic cancers only affect one breast, but in some cases these develop bilateral breast cancer. NF2 gene results in bilateral acoustic neuroma. That's a hereditary tumour syndrome, it's not a cancer syndrome. defective p53, so if you inherit a defective copy of p53 from one of your parents, you'll end up with multiple tumours at multiple sites, so multiple independent primary cancers. Defective HMLH1 or HMSH2, these are DNA repair proteins, so if you get mutations in those inherited from a parent, you get this hereditary non-polyposis colorectal cancer, HNPCC. So these are all hereditary cancer syndromes, and by studying these patients and finding out which gene is defective has allowed us to identify some of the major tumour suppressor genes. Now, another major tumour suppressor gene is the RB1 gene, which codes the PRB protein, and this also causes a hereditary cancer called retinoblastoma, but it comes in two forms. You've got your hereditary form and your sporadic form. The hereditary form, obviously you need a parent who has got retinoblastoma, so for tumour to occur, people are born with one copy of the RB1 gene which has become inactivated, and the second copy is inactivated by somatic mutation. Exactly the same situation as here, where they've got the APC gene. First hit can be germline mutation, second hit can be a somatic mutation. So, because there's two mutations required, we call it the two-hit model, and it was proposed by Knudson in 1971, and this is before the concept of tumour suppressor genes was really known about. And if you click on this link here, you get a really good explanation of Knudson's hypothesis and how he came to his conclusion. But basically what Knudson did is looked at hereditary retinoblastomas versus sporadic retinoblastomas and realised that patients with hereditary forms presented earlier and often had bilateral disease, whereas other patients had no family history and they always presented with sporadic forms of the disease. So let's take these two situations. If you're born with a hereditary form of retinoblastoma, this is a diagram that purely illustrates chromosome 13, which is where the RB1 gene is, and an X represents a mutation in the retinoblastoma gene. So this cell here could be, the cell at conception, and as that cell divides, gives rise to more and more cells. Any one of those cells can pick up a second mutation of the RB1 gene, and that can form as a tumour. Now, as it is, there are very few cells that are completely and utterly dependent upon RB1 in this way, and some of those cells are the developing cells of the optic nerve and retina. So if one of these cells develops a second mutation in RB1, that will turn into a tumour cell. Compare that with a person who is conceived normally for RB1. At conception, every cell is normal for RB1. There may be one cell will pick up a mutation in RB1. That will give rise to thousands and thousands of other cells which have also got one mutation of RB1, and any one of those could then pick up a second mutation and develop into a tumour. Now what we find is that if a person is born with this mutation in every single cell, then this is very, very likely to happen. It's going to happen in almost every patient. However, if a patient is born normal for RB1, this event of two mutations in the same cell is incredibly rare, and in retinoblastoma it's about 1 in 30,000 live births. So this shows a very brief pedigree of a patient and family with retinoblastoma. So squares are male, circles are females, and what it shows here is details of each individual case. So a solid filled square is a patient who is clinically being examined and shown to have got retinoblastoma. So that's a male with retinoblastoma, that's a female with retinoblastoma, and in this particular case, if the square or circle is fully filled in, that's showing bilateral disease, and if it's half filled in this particular case, it's showing the tumour in one eye but not the other. So what we can see from this is this female here was the person who had retinoblastoma. We know that that was diagnosed at 4 months. 4 out of 12 is 4 months. and that question mark is we don't know about the other eye at all, presumably because the patient can't be clinically examined. This patient, we know that they presented with at 3 months and at 6 months, and their offspring, with someone who was obviously not retinoblastoma positive, is presented with tumours at tumour in each eye at different time points, and the sibling has also presented with at least one tumour in one eye. These numbers underneath, 34 is the age, so this is a 5-year-old who's had 2 tumours develop in the first year of life. This is a 2-year-old who has 1 tumour presented at 8 months and then no tumour yet. So this patient is 2 years old, and so many patients do develop bilateral disease, some patients develop unilateral disease. So this is a patient that's, been slightly more lucky than the sibling. Here's another much larger pedigree. This is a pedigree for the mutation in the APC gene, which results in familial Adenomatous Polyposis Coli, FAP. And so in this we've got multiple generations all showing a cancer predisposition. And we can track back, and one of these 2 up here would have been the original source of the mutation. And we know that because multiple offspring from this couple have got stomach cancer, which is a feature of APC as well, colon cancer and colon cancer. So if you've got multiple individuals, then one of these will have had that as well. And then we look at different lineages down here. You can see here this person died of lung cancer without diagnosis of any colon cancer, but their offspring have also got polyposis, that's multiple polyps, and some of those have got cancer. And you can see down here you've got individuals with polyps in their colon, which is caused by this gene. So the polyps are the precancerous stage, and then they will sometimes develop into fully fledged cancer. What we can see is this particular person here made it to the age of 70, died of lung cancer, but the colon cancer either never developed or never developed in a clinically relevant way. So this patient could have had lots of colon polyps, you just wouldn't know because this was someone quite a long time ago. And with better screening, you can see that patients are getting diagnosed very early. So we'll start to screen adults for polyps in their colon if they have a family history of this particular disease. So in cancer genetics, in genetics in general, we use the term penetrance. Penetrance means how likely it is you're going to get the disease if you got the mutation. So something that's highly penetrant means that if you've got the mutation, you're going to get the disease. Now this person here has avoided clinical manifestations of the APC gene. So, APC may be 90-95% penetrant for cancer, or at least polyposis. But you only know about the polyposis if you go looking for it, or it presents as symptoms. So all of these patients who have inherited a defective tumour suppressor gene have undergone Knudson's 2-hit hypothesis. That is, they've inherited one mutant copy of one of those genes and then taken on a somatic mutation in the other copy of that gene. And that somatic mutation can be one of many different forms. It could be a point mutation, it could be silencing of the promoter of that gene, it could be that the entire gene has been deleted by either partial or total chromosomal deletion. Now the next main genetic concept that I'm going to cover is the mutator phenotype. So we've seen on the multi-hit models that you get an initial mutation such as loss of APC, and then a second loss of APC, and then we have a cluster of cells which forms an atypical crypt within the colon. The question is, how do we get from that to a fully fledged cancer with millions of mutations throughout the genome? And the reason why that happens is we eventually get a mutation that causes the mutator phenotype. The mutator phenotype is where cancer cells acquire mutations at a much higher rate than normal cells, and that allows mutations to occur very, very quickly. In colon cancer, we tend to see a phenomenon called microsatellite instability, where the DNA mismatch repair enzymes, HMLH1 and HMSH2, fail to repair single base pair change mutations, and therefore the tumour cells rapidly accumulate mutations. We can also see chromosome instability, where DNA repair processes that spot chromosome abnormalities, unequal chromosomal passing to mitosis. So in some situations you can have an additional chromosome passed to a daughter cell. That will be part of chromosome instability, but chromosome instability can take many forms. I'll show you some of those later on. So basically these can contribute to the cancer cell having vast numbers of mutations. And a key point is that lack of genome integrity is the underlying cause of carcinogenesis that allows those multi-hit models to accumulate all of the mutations required for cancer formation. Okay, so the first one of these mutator phenotype genetic concepts that I'm going to cover is microsatellite instability. Microsatellite instability is where short repetitive sequences of DNA are poorly copied by the tumour because they have a defective DNA mismatch repair enzyme, typically HMLH1 or HMSH2. So these repeats of DNA, TA repeats and CAG repeats, are very difficult for DNA polymerase to copy without making a mistake. If the DNA mismatch repair enzyme is defective, you get widespread misreading of these sequences and lots of mutations. Now these are not quite so crucial for affecting gene expression or anything like that. It's just that the same enzymes that repair mismatches in these also repair single base pair mutations, and it's the single base pair mutations which seem to drive carcinogenesis. It's just that we can observe microsatellite instability very easily by doing polymerase chain reaction analysis of microsatellite regions of DNA and analysing them on the gel to see whether a tumour is MSI positive, microsatellite stability positive or not. And this is shown in this particular image here. This is a study that I did many years ago. And what we can see is N is normal, T is tumour from the same patient. So N is from blood DNA, T is from tumour DNA. And all we do, we extract that DNA and we perform polymerase chain reaction across a microsatellite region. Now some of these microsatellite regions are what we call polymorphic. You have two alleles, one from your mother, one from your father. They're of different sizes and we can see the difference between them. So there's a small allele, there's a large allele, and this could be is likely to be 2, 4, 6, 8, 10. So this allele is 10 base pairs smaller than this one, and these shadow bands here are 2 base pairs apart because it's a TA repeat. So what we can see, normal and tumour, they've both got a small allele, so therefore nothing has happened here. In this particular tumour, there's a large allele here in the blood, a small allele in the blood, and when we look at the tumour, this small allele is still there at the same size, but this larger allele here has shifted. Now the reason this has shifted is because the tumour cell is defective in a mismatch repair enzyme and has made a mistake and has shifted this allele by 4 base pairs. So this is what we call microsatellite instability. And what we do is we compare normal tumour DNA, so normal DNA to normal blood DNA from the same patient, and analyse them for 10 microsatellite markers, and if we see any shift of any of those, then that will signify that those tumours are microsatellite unstable. So it's a simple DNA test that we can do in the lab to identify microsatellite instability. Now the microsatellites themselves are not coding DNA, they're usually not very important. They're just a signifier that this effect is going on. However, what is really important is where you've got runs of single sort of mononucleotide tracts, where you've got a run of, in this case, 8 Gs or 8 Cs or 10 As. And these are where base strand slippage occurs, and the mismatch repair system corrects mistakes when you get mismatches here. So If DNA polymerase is copying this DNA and makes a mistake and puts in an extra G, there will be a mismatch. And one of these Gs will be not paired up with a C on the opposite strand, and there'll be a mismatch. The mismatch repair system will spot that and correct the error. However, if the mismatch repair enzymes are defective, this extra base pair that has been put in will be copied and be present in all of subsequent daughter cells. Now, the Bax gene has got a mononucleotide tract, and it's very frequent in these mismatch repair positive tumours to have an extra base added in at this mononucleotide tract, and that renders the Bax gene inactive because the reading frame has been shifted. Similarly, the TGF-beta receptor type 2, which is a antiproliferative growth factor receptor, that has got a mononucleotide tract of 10 A's, and you often get either loss of a base or gain of a base, and therefore that will render TGF-beta receptor inactive. So this is actually the pathological effect of a mismatch repair defect. The actual shifting of microsatellites is completely irrelevant. It's just an easy to spot visual or analyzable effect that we can observe. Okay, so microsatellite instability is all about genetic defects on the molecular level at the single base pair level, whereas chromosome instability is looking at gross chromosomal abnormalities, and the fundamental reason for that is often very different. This is some chromosome instability work that I've done, again, many years ago, and it's showing what can happen within tumour cells. So this is a relatively stable partial karyotype. And when I say a partial karyotype, these are the only abnormal chromosomes in a particular tumour cell. And we can see chromosome 5 with a bit of chromosome 11 on the bottom. And then there's a chromosome 17 to 20 translocation going on between these two. Okay, so that's relatively simple. However, in some tumour cells, we find these, which are hugely complicated combinations of multiple individual chromosomes, and where we get chromosomal fragmentation and then recombination, we get very rapid emergence of new phenotypes. So in this particular study that I did here, what we showed is that if we selected out the most invasive cells from a population of cells, they tended to have highly unstable chromosomes, and those chromosomes were sort of signifying that these tumour cells, the chromosomes were breaking apart, sticking back together and creating new phenotypes. Some of those phenotypes were highly invasive, and that's what we were selecting for. And this individual chromosome here is a 4 and a 5 stuck together at the base. This we think is made of chromosome 15, a bit of chromosome 14 and a bit of something unknown in the middle. So this is massive chromosomal genetic damage driving new phenotypes, and the same thing happens within tumours. Okay, so here's the original karyotype of one of my cell lines. This is a uveal melanoma cell line showing not an awful lot of structural abnormalities, a few additional chromosomes here and there, one isochromosome. So this is what the cell line started off like, and if we put that through an invasion assay several times, this is what, and then selected the most invasive cells, this is what it ended up looking like. And what you can see is we've selected for cells which have got massive chromosomal abnormalities. So if a cell gains a new ability such as invasion, that gives the cell the ability to leave the primary tumour site and then colonise a different site. So what we often find is that the chromosomal changes in a metastasis are far more complex than what they are in the primary tumour. And what we managed to do is model that in a cell culture assay. So chromosome instability is a visible phenomenon. We can do a karyotype of the cell and look at the chromosomes, and we see occasionally very chromosomally unstable cells. These often seem to be selected for in, these populations are selected for in metastases, and you end up with more complicated chromosome abnormalities in the metastasis than the primary tumour. There are multiple mechanisms by which huge levels of chromosome instability can occur. One of them is breakage-fusion bridge events, one of them is chromothripsis, so we'll have a look at those. So breakage-fusion bridge events are what was occurring in my study on uveal melanomas many years ago, because one thing that I noticed in this particular study is we got lots of telomere associations. So this is where the telomere of one chromosome sticks to the telomere of another chromosome, and you end up with what's called a dicentric chromosome. Now, that in itself, those chromosomes are defective and there are probably genes missing and damaged at the fusion site here and here. However, this becomes particularly problematic when this cell tries to undergo go through mitosis, because you can end up with spindle fibers attaching here and here, and spindle fibers attaching here and here, and that should separate those two chromatids. However, because you've got two centromeres, you can have those spindle fibers attaching and basically ripping the chromosome to pieces. And if you follow this link here, you'll find a paper on this. They form initially what's called an anaphase bridge. And so you get a situation like this, where this is the dicentric chromosome, and in red is one chromatid, in white is another chromatid, and then they try to undergo an abnormal anaphase, because of where the mitotic spindle is attached, those chromosomes get ripped apart, and then the chromosomes, as an attempt, the cell attempts to resolve the issue by sticking those chromosomes back together and carrying on. Now, you have to remember that a normal cell should die at this point, but the cancer cells often have defects in apoptosis and survive this process. So we see this thing called breakage-fusion bridges leading to anaphase bridges, and these are chromosomes that are attached to the mitotic spindles in one cell and still attached to one in the other cell. And as cytokinesis occurs, those chromosomes get ripped down the middle, but then the cell just carries on regardless. And this drives huge heterogeneity within cells that undergo breakage-fusion bridge events, and you end up with, you see this phenomenon with lots of dicentric chromosomes and then lots of fragmented chromosomes, and then you get seemingly random chromosomal rearrangements. This leads to new phenotypes, some of which will be highly invasive cells. And this particular article here suggests that breakage-fusion bridge events may actually be responsible for oncogene amplifications, in this case the ERB2 or HER2 gene amplifications on chromosome 17. And what we see on that is we often get back-to-back tandem duplications of the ERB2 gene, and this is a fluorescence in situ hybridization showing multiple copies of the HER2 gene in breast cancer, and if there's multiple copies, those patients are treated with Herceptin. However, the phenomenon behind it could be down to breakage-fusion bridge events, where you get breaks and then fusion of chromosomes and then breaking again, and basically this duplicated DNA at this event here, so can then become copied and copied and copied again, and you end up with a increased gene copy number of the ERB2 gene. Now, as a cell develops additional copies of the ERB2 gene, that makes that cell more proliferative in this particular case, and therefore that particular clone of cells will grow more quickly. So in this situation, it's selecting for a gene amplification that gives the tumour some sort of benefit. Another genetic phenomenon that can result in chromosomal instability is what we call chromothripsis, and this is where you get sudden massive chromosomal damage due to a single event. This could be a passing alpha particle, a radiation particle, hitting a chromosome almost end on and causing catastrophic chromosomal damage, and then all of these fragments of DNA get randomly shoved back together by the tumour cell. Remember, the tumour cell should die at this point, but it doesn't because of its lack of apoptotic pathways. So you end up with a hugely complicated chromosome. So the chromosome starts off, this is the normal chromosome, it gets fragmented into multiple pieces, they get stuck back together, some bits get lost, and then this new chromosome here, the combination of the loss of this region and the rearranging of this region helps to progress the cancer towards, along that mutator phenotype of accumulation of multiple mutations. And this review summarises the concept quite nicely, and it's shown with individual chromosomes that were designated, split up into different regions, and showed that when they underwent chromothripsis, you could get them completely recombined, sometimes back to front, sometimes completely reordered, some bits deleted, some bits that didn't contain a proper centromere form these things called extra chromosomal elements. They can be ring chromosomes, or they can be double minute chromosomes, which are just extra chromosomal elements. These can then become easily amplified if they contain an oncogene sequence. These regions that tend to be preferentially lost are the locations of tumour suppressor genes, and then here we can get fusion genes. If you think about TMPRSS2 ERG in prostate cancer, sort of translocations and fusion genes. So all of these can combine to give huge amounts of genetic heterogeneity within a tumour, driving the mutator phenotype. Okay, so that's the end of this particular recorded lecture. So what we've effectively covered here is multiple genetic concepts that are leading to driving chromosomal and genetic heterogeneity, and that leads to phenotypic heterogeneity. And if you think right back to the very start of the session, I was talking about the monoclonal versus the polyclonal nature of cancers. And everything that I've just told you here helps to explain why tumours appear highly heterogeneous, yet they can still have evolved from a single cell. So that's the journey that we've been on, of going from a single cell, accumulating a single mutation of a tumour suppressor gene, and then all of the events that can occur right up until having a highly invasive, highly proliferative metastatic cancer.